Hello everyone! Today we'll explore AMG 329, a promising antibody for autoimmune and inflammatory diseases. We'll cover its background, design, and key properties, target, development, preclinical data, and clinical potential, etc. The FLT3 receptor and its ligand, FLT3L, constitute a fundamental signaling axis in immune homeostasis. This pathway critically regulates the development, survival, and functional activation of dendritic cells, which are central orchestrators of immune responses. In several autoimmune conditions such as rheumatoid arthritis, Sjögren's syndrome, and systemic lupus erythematosus, elevated levels of FLT3L have been observed, correlating with increased dendritic cell populations and enhanced inflammatory activity. Therefore, pharmacologically interrupting this signaling cascade offers a rational strategy to modulate pathogenic immune activation while preserving broader immune competence. FLT3 expression is tightly regulated and predominantly found on specific immune cell lineages. It is constitutively expressed on hematopoietic progenitors and maintained on key dendritic cell subsets, plasmacytoid DCs or PDS, which are prolific producers of type I interferons. Conventional DCs, including CDC1 and CDC2 subtypes, and a subset of classical monocytes. Binding of FLT3L to its receptor triggers intracellular signaling that promotes cell proliferation, survival, and the acquisition of pro-inflammatory functions. In autoimmune settings, this pathway becomes overactive, contributing to the persistence and hyperactivity of these immune cells, thereby sustaining inflammation and tissue damage. The journey of AMG 329 began with the isolation of a parent antibody clone, designated 5D9, from a human phage display library. This clone exhibited cross-reactivity with human and cynomolgus monkey FLT3L. To enhance its therapeutic potential, the antibody underwent affinity maturation, yielding a high-affinity variant known as M40. This optimized binding domain was then grafted onto a human IgG1 backbone that had been engineered with 3 specific point mutations, L234F, L235E, and P331S in its FEC region. These mutations effectively silence FEC-mediated effector functions. The final molecule, AMG 329, was rigorously validated using biolayer interferometry and high-throughput screening assays, confirming its potent ability to block the interaction between FLT3L and its receptor. In drug development, understanding which species an antibody works in is vital. AMG 329 binds strongly to human and cynomolgus monkey FLT3L, which are 95% identical. However, it does not bind to mouse or rat FLT3L, which are only 73% similar to human. This means preclinical testing can be done in monkeys, but not in standard mouse models. The main job of AMG 329 is to block FLT3L. Lab tests show it neutralizes both free FLT3L in blood and FLT3L attached to cells. Importantly, it does not affect similar signaling proteins like stem cell factor or macrophage colony stimulating factor. This high specificity helps avoid unwanted side effects. AMG 329's safety is enhanced by its engineered FEC region. Special mutations prevent it from triggering immune cell killing ADKI or activating the complement system CDC. However, it still binds normally to the FEC-RN receptor, which is important for maintaining its long circulation time in the body. In monkey studies, AMG 329 reduced key immune cells including plasmacytoid DCs, conventional DCs, and monocytes in a dose-dependent way. These effects lasted during treatment and were fully reversible after stopping. The antibody also showed a stable pharmacokinetic profile, supporting dosing every 2 weeks in clinical trials. To test whether AMG 329 weakens overall immunity, treated monkeys were given clay-AH, a protein that normally triggers a strong antibody response. Treated animals produced normal levels of anti-clay-AH antibodies, just like untreated controls. This shows that AMG 329 specifically modulates the immune system without causing broad immunosuppression. More target sharing. See you next time.